Hi, I'm Santamaria, and today I'm going to be talking about the knockdown of NA25 and how it suppresses breast cancer progression by regulating apoptosis and the cell cycle. So, what's breast cancer? It's a disease in which the cells in the breast go out of control. It's mostly diagnosed in women, and it can start in different parts of the breast, such as the nipples or the ducts. And the most common symptoms are change in size in one or both breasts, lump or swelling in armpits, dimpling of the skin, and rash on or around the nipple. What's NA25? It's a protein-coding gene that appears to be very prevalent in breast cancer. But when it's deficit in cells, there's an increase in cell apoptosis, delays in G2 and M phase, and decreases in cell clone formation. So hopefully in the future, the knockdown of NA25 can become a new form of therapy for patients with breast cancer. So the objective. We're trying to see the relationship between NA25 and breast cancer by comparing the gene expression between normal tissue and breast cancer tissue. We're specifically trying to see the effects that the gene has on breast cancer cells when it's altered, knockdown. Methods. Western blot, what is it? It's a technique that is used when wanting to detect a specific protein in a blood or protein sample. We normally use gel electrophoresis in this technique. And colony formation assay, it's an in vitro cell survival analysis based on the ability of a single cell to grow into a colony. Some steps in this technique are treatment, plating, and staining. As we can see here, an example. Apoptosis assay detects and quantifies how many cells have undergone apoptosis, which is programmed cell death. And RT-qPCR monitors the amplification of a specific DNA molecule during PCR, which is polymerase chain reaction. Results. So we could see the usage of Western blot and RT-qPCR to analyze the NA25 expression in breast cancer cells and paracarcinoma cells. So we can see that the NA25 was highly expressed in breast cancer tissue, and also over here, the thicker the line, the more expression. Over here in A, we can see that NA25 gene expression in breast cancer cell line T47 was far greater than in just normal tissue MCF10A. Over here in B, after the knockdown of NAA25 and T47D, the mRNA and protein expression of the gene was diminished. Now a discussion. These first batch of authors said that the INCH SAT164 was highly expressed in breast cancer, and it is shown that when overexpressed, it could promote colony formation, and when downexpressed, it could promote cell apoptosis and regulation of the cell cycle, which the article agrees with. The second batch of authors said that the highly expressed IFIT2 and NDRG1 could reduce tumor migration and metastasis because NA25 knockdown could upregulate IFIT2 and NDRG1 expression, which the article also agrees with. Lastly, these batch of authors said that as an important accessory subunit of the NATB enzymatic complex, NA25 could work with the NA20 catalytic subunit to promote enzymatic activity, which the article also agrees with. Lastly, conclusions. First, molecular biology gives the medical field different methods and techniques to learn and see how different diseases are created and how they progress. Without these tools, we as doctors wouldn't be able to come up with effective vaccines or treatments to stop these illnesses. Lastly, investigation in the medical field is heavily based on molecular biology, since it's important to know how cells function, which cells are in charge of specific reactions, and what happens when a cell has an error in their genetic code. This helps us understand how diseases work and hopefully how to prevent them in the future. Thank you.